Welcome back, everyone. Next, we have BioVaxys Technology Corp. It trades on the OTCQB under the symbol BVAXF and on the CSE under the symbol BIOV. It's a clinical stage biopharmaceutical company dedicated to improving patients' lives with novel immunotherapies. Please welcome its CEO, James Passin. CEO and President, Ken Coven, and CEO and President, CEO James Passin. Welcome, gentlemen. How are you today?
Good morning.
All right.
Great.
So
Good.
there has been a lot of news lately about BioVaxys and your DPX platform. It's a breakthrough technology in the immuno-oncology field. So share with us, what exactly is DPX?
Sure. Thanks, Anna. Let me move ahead to the first slide here. DPX is a fundamentally disruptive antigen delivery system. It's entirely different than what's currently available on the market now. DPX is a non-aqueous, it's not an emulsion, non-systemic delivery system. And the mechanism of it basically is that the lipid nanoparticle that is DPS is injected under the skin, containing a cargo of the antigen that we would like to have distributed to the target. And that antigen, the antigen that's in DPX, is engulfed by APCs or antigen presenting cells, which is how this naturally, the immune system naturally works in the body. The APCs then transit to the lymph node and trigger the release of immune cells such as B cells and T cells that actually do all the heavy lifting against whether it's a tumor antigen that you're targeting or an antigen on a viral surface. We like to look at DPX as really it's like a giant truck, and we can put in DPX basically any kind of cargo that we wish to, whether it's a peptide, a protein, a polynucleotide, mRNA, small molecules, whatever it is that is the antigen, we can place inside of DPX and use that to trigger stimulate a very robust immune response. It's very flexible too, in that it's like a plug and play format. We can drop in different antigens, as I said, and we can use it to address immune system disorders, whether they're viral infections, whether it's cancer, whether it's immunodisorders like food allergies, for example. Whatever it is that you need to be able to deliver to have an immune response, we can pack inside of DPX.
Wonderful. And BioVaxys and its DPX platform are in the news again today. Tell us a little bit about this new study that's being presented today, where you are at the Personalized Cancer Vaccine Conference in Boston. We can see Boston behind you. It looks beautiful and cold.
It looks beautiful, cold, and snowy. So I'll add to that. But the study that we're presenting today at the conference was done in collaboration with Dr. Hakima Nick, and she's at the Ohio State University Comprehensive Cancer Center. And it's actually a collection of different animal studies that were done to evaluate DPX. The first study that was done was a murine or mouse therapy model. And what was done is in one group, mice were implanted with tumor cells, so they would get cancer. 7 days later, the mice in that one group were receiving injections of DPX plus something called R9F. And that's as I said, you package antigens. That's a antigen from HPV, human papillomavirus, that's typically used in these kinds of studies. The second group of mice, they were also implanted with tumors, so they would develop cancer. They were then injected subsequently with CD8 CD8 positive T cells. These are cytotoxic T cells. They're killer cells, more or less, and that's what you want to see triggered, to help combat different tumor types. After the animals were injected, with CD8 T cells, The donor mice, or the mice that received that, were then treated with DPX-R9F or untreated. And what we saw in that particular case was that DPX formulations that had tumor-derived peptides delivered are highly effective vaccines to inhibit and prevent the growth of tumors. This is more or less done as a baseline for the study. We've seen this. This has been verified in a number of clinical studies as well too. But we wanted to establish it as a baseline for us in the study. Second, what we did was let me go ahead here to whoopsie. Okay. Let me put this graph up here. Thanks. Okay, so what I wanted to show here is just the results of this, and it's a very nice graph if you want to bear with me for 1 second here. The first graph on the left-hand side, the purple is DPX and RD9F treated, and you can see that by study day 20 in the purple at the bottom, there was barely any growth at all in the size of the tumors following the challenge in the mice. On the right-hand side, these are the animals that were treated first with CD8 positive T cells, and then we looked at whether these animals, if they were treated with nothing at that point, just the CD8 cells, or with DPX and R9F, and you can see again that out to like past 20 days of therapy, that the tumor volume really did not increase very much. So this is really positive and a great endorsement of what DPX can do when packaged with the appropriate antigens.
And with
The
that,
next study,
yeah.
I'm sorry, go ahead.
No, you're going on to the next study. Go on.
Okay. The next study that was done was a what's called a METH-A animal tumor model. And in this case, we looked at injecting DPX with Neo1, which is a very common rejection peptide. It's a neoantigen, and that's really becoming now the more or less the holy grail in cancer center therapy to develop appropriate neoantigens. So in this case, we packaged a neoantigen in DPX, and we included with that a drug called 9D9, which is a checkpoint inhibitor. No explanation needed for those. What we found from this is that DPX packaging this neoantigen, in addition to the 9D9 checkpoint inhibitor, was found to be as effective in preventing tumor growth as what is really the gold standard in this case, which is the neoantigen plus dendritic cells. So this was really exciting for us to be able to see this, that we matched the efficacy in this animal model with what's currently seen as the the gold standard. And in the third study that was done, this more or less was the the icing on the cake for us, is that we took DPX and added TFDPA, which is a rejection peptide, and that showed amazing results in the animal model. What we saw was that this particular DPX peptide formulation performed way better than TFDPA plus the dendritic cell vaccine. So before we showed parity with the gold standard, now we're showing superiority against it. And what's really interesting from this and of extreme importance to us commercially is that these experiments with DPX showed that either by packaging an antigen plus 9D9 or not packaging any kind of an antigen, we showed that DPX on its own had very significant immunoprotecting effects. And this is really exciting for us. It potentially opens the door for looking at DPX as an adjuvant, and that's a huge market potential there. Or else also looking at DPX in combination with eticargo with checkpoint inhibitors, and that's really exciting because we've already proven that DPX in safety and tolerability studies, and have a number of INDs already done or filed for that. That we can look at DPX with the checkpoint inhibitor as a possible therapeutic.
And you also mentioned in your PR the DPX adjuvant studies. So if you can explain that a little bit more.
Yes, sorry, I think I went ahead a little too fast on the slides.
Okay.
But
well, with all of this, yes.
Yeah, no, I did. I covered that in the other slide. So, in conclusion, what we saw from the studies that were done by Dr. Nick is that the DPS formulations, they're very effective, in challenging, tumor growth, with a cargo peptide, as we knew. DPX formulations are effective with the HPV peptide that was onboarded and neoepitopes that were derived from the meth-A tumors. And that bodes really well for us in terms of developing neoantigens packaging DPX. We also saw that DPX formulations were a lot more effective than commonly used adjuvants. And there are about 4 or 5 different adjuvants that were used in the study. These were very typical adjuvants that are combined with currently existing marketed vaccines, and shows that mixing DPX with these, it was actually superior as an adjuvant compared to these. DPX we saw was more effective, or as effective as bone marrow dendritic cells, and as I said, that's the gold standard. But we also saw that with the different antigen in there that we studied, that it was actually superior to dendritic cells. And of course, finally, that DPX on its own, without any kind of cargo peptide, has a very, very meaningful immune stimulating property to it.
And so, with all these new findings, what does all this mean for your company?
Well, good stuff. It's certainly not only an endorsement of the the DPX platform that we acquired earlier this year, but it opens the door for further study of DPX, either on its own as an adjuvant. It opens the door to further study of DPX in combination on its own with a checkpoint inhibitor, and it also further verifies what we knew at this point, that DPX plus a particular antigen can stimulate a very powerful immune response. And from a scientific perspective, the data is really exciting. From a commercial perspective, it's extremely intriguing for us, because it looks like it can open some new doors.
And with all of this, what is BioVax's overall plan for developing its pipeline?
Yeah, James, did you want to tackle this? Or?
Yeah, Ken, absolutely. Great presentation, Ken. So, look, in terms of our plan for next year, we are looking for opportunities to license our lead products. So we've got an initiative we started with that. We also, once we lock down the supply of 43 kilos of GMP grade lipids, we will have enough raw material to really support any preclinical or clinical trial in the next several years. We have very constructive engagements with the principal investigators that were involved in many of these uncompleted phase 1 trials. There's just an interest there to resume. And we also intend to initiate preclinical work on both DPX mRNA as well as our allergy desensitization.
And as we are coming into 2025, what are some milestones that you're expecting?
James, you want to,
Ken,
you want to do that?
Do you want to walk through some of the scientific milestones?
Say again, you got cut off there.
Yeah, do you want to address some of the milestones in terms of our research initiative priorities?
Oh, yeah, sure. Yeah, sure. We have a number of milestones that we're looking forward to for the upcoming year, both on the internal development perspective, and also in terms of further building out the company. From the scientific side, product development space, we're looking at initiating our preclinical study at McMaster University on DPX packaged peanut antigens for prophylaxis for peanut allergy. And that's a really exciting project for us because there really are no good, protective alternative available for people with severe life-threatening peanut allergy. We're looking on moving forward with a team of researchers at Dalhousie University that have expressed a lot of interest in working with DPX in the oncology space, and we're currently in the process of designing various studies and protocols that would go along with that. We're also looking at restarting some of the studies that unfortunately were terminated during the IMV bankruptcy, and one in particular that we're looking at is a study that was ongoing at Laval University in bladder cancer that looked at DPX packaging survivins, the survivin proteins, as well as a novel protein called MAGE9. And the preclinical work in that was excellent, I guess is the best way to put it. We're hoping to get a publication on that soon with the from the researchers at Dal. But that particular study had to be terminated after only a handful of patients were done because of the bankruptcy. And given the preclinical study and how exciting that was, we want to go back and take a look at that because the results that we saw were tolerability was good, but it was kind of indeterminate on the immunological side of that. So that's another project that we're looking to kick off in the upcoming year, and we're planning with that now. Of course, we're looking to continue the studies that Dr. Nick had done on DPX, and that's in the planning stage, as well, also. So I don't know if I missed anything with that, but
Ken, we should also mention we should also mention our two licensees in animal healthcare, Zoetis and Spayvac. Both will go in for respective regulatory approval next year, so we might see significant progress in terms of our potential revenue-generated licenses.
Great, thanks for mentioning that. That's important, for us because we're, those are going to be revenue generators for BioVaxys, and both companies are doing a bang-up job in terms of preparing for filing and eventual launches. And I think you may have seen some press releases we put out recently on Spayvac and how they've been really ramping up their production capability.
Fantastic. We have some questions from our viewers. Clark is asking, how much of this technology is proprietary?
That's a broad question. The easy answer is to say all of it. The patent portfolio that we have includes DPX, the product itself. It includes methods of manufacturing, methods of production, methods of use or utility. It includes various applications or issued patents on the DPX plus various antigens. So all of it is proprietary. We acquired, I want to say it's 21 families of patents and close to about 120 applications or issued patents, from national stage filings. So it's a pretty big and aggressive portfolio. And it is all proprietary.
And regarding the use of a market maker, does BIOV still use Maxim Group? And do you have any option regarding the relationship between the stock price and financing?
Yeah, I can address that. So Maxim was previously the advisor to BioVaxys with a number of different responsibilities in that relationship. Both parties agreed to terminate that relationship. The shares issued to Maxim were canceled. So we don't have any involvement with Maxim at this point, although they are a great life sciences focused Wall Street Bank. So we're happy to have a relationship with them. Currently, we're not doing anything with Maxim. We are working with another market maker called ITG.
Great. And a question from Jordan, if you can expand a little on the relationship you have with Zoetis. Is this relationship you see expanding or remaining as is?
Jordan, that's a really good question. The original license agreement that was done with IMD and Zoetis, that was for developing in Australia and Brazil immunocontraceptive vaccines for hoofed breeding stock. And there may be some interest in expanding that. Spavac has been extremely aggressive in doing new studies with aquaculture, for example, and some other fields that were included in the license agreement with them. The Zoetis agreement was for hoofed stock, and there's always a very good possibility that we would expand that. In speaking with Zoetis, we know that there is a very large, promising market in that application for immunocontraceptive vaccines. That really is going to come down to their decision as to whether it's not a question of whether the vaccine, whether it works. It's a question of, is there a commercial application, in those markets, for it. And we may very well expand that as time goes on, but we'll see where that goes.
And have you ever had a plan to discuss with some government level entities, maybe talking about grants or support? Jordan also asks.
Yeah, actually, most recently, we were just invited to join a consortium of big pharma, CDMOs, academic institutions, government labs that are all part of a group that's supported by BARDA. And the focus on that is for vaccine work. And we've actually been contacted and have contacted groups in the consortium that are interested in putting together a proposal for funding by BARDA. And there is an open solicitation out for that now. So, we're looking at that. And it's a matter of, putting together capabilities on the delivery side, which is us at BioVaxys with DPX. We're looking at, in this case, the study is with influenza. We're looking at collecting the antigens, mRNA antigens for influenza, and there are collaborators in that group that have that. So they would contribute that particular piece. There are other collaborators that specialize on different flu models, and that's the piece that they would put in. So it's a question of mixing and matching all the different groups that are interested in the project and have the capabilities. So we're looking to try to move that forward right now, for that particular open solicitation. As far as grants go, we're looking at grant opportunities in the U.S. and in Canada, and in particular, one that's comes top of mind right now is we're working with some expertise in Canada on the grant side, and they're in the process of connecting us with different agencies and within the Canadian, the provincial governments to execute a grant for hopefully for the phase 1 study of the peanut allergy program. So we're looking ahead now at this point with the phase 1 study for that.
And how is the current BioVaxys program superior to other developers of similar tech out there?
We look at this, as I said early on, we look at this as it's a fundamentally disruptive technology. I know that sounds like buzzwords, but if you look at it, it really is. I mean, this is a case where the platform is the product, for us that we're developing, and we recently had shared some data where we looked at comparing DPX to the most common, antigen delivery mechanisms, which are aqueous formulations and some oil-based formulations. And what's different and unique and superior about DPX is it's non-systemic, and that first and foremost is probably one of the most critical aspects of it, because what that means is that whatever we package in the body stays right where you put it, like under the skin. And that means that gives the ability for the APCs, the antigen presenting cells, to engulf that, pick it up, and take it to the lymph. When you're using an aqueous delivery system or an emulsion like an oil-based, that doesn't happen. Instead, what you've got is systemic or wide circulating aqueous formulations or emulsions that really deliver the antigen anywhere. And that actually has been one of the problems facing like mRNA delivery, and using lipid nanoparticles. Yeah, they can package it and do a nice job doing it, but it's systemic, and that opens the door for a lot of off-targeting. And in particular with packaging mRNAs, we think that using our approach is going to be better because it is a non-systemic approach. So it truly is different and unique than anything that's out there. And thinking back of what we said earlier about what we presented in Boston, we're presenting in Boston today. Here's a technology that works on its own and stimulating an immune response, and you certainly don't see that with an aqueous formulation. And that's something that we had shared, in a study, in a press release a couple of weeks ago, on the differences between DPX versus these other formulations.
Great. Thank you for clarifying that. One more question from Edward. Once it's more of a follow-up question, if you can elaborate a little bit more on your capital structure and potentially uplisting to a national exchange.
Yeah, I'll address that. In terms of our cap structure, we've got about 267 million shares outstanding. The stock's actually reasonably tightly held. 45% of shareholders voted in the recent shareholder meeting, which showing a lot of engagement with the stock. We absolutely eventually want to uplist. I'd say right now it might be difficult to meet the requirements for NASDAQ, but eventually we will get to a higher tier exchange, and it's a matter of increasing the market cap of our float and meeting the other requirements.
Wonderful. All right. Well, do you have any closing remarks for our viewers today?
Yeah, I think the news today really highlights the revolutionary nature of DPX. We are very excited to move forward with all of the DPX products. And there's no other, we're not aware of any other lipid-based antigen delivering platform that is non-systemic. So we really think there's a lot of new inroads that we can make with this technology.
And just add, if you have the option, don't try to come to Boston today. The weather's not very nice.
It looks pretty though. All right, gentlemen, thank you so much for your time and presentation, important work you're doing, and we look forward to seeing you in the new year.
Thanks so much, Anna.
All right, stay with us, everyone. We'll be right back.